I've been accepting them.
You have been accepting them. Okay, right. That's good. Matomo, do you see the chat? I see you.
Yes, I do see the chat. I suspect it's available to those who were invited, but not the guests. But I'm going to set it now. Let me just add presenters, then we can
Okay.
hit the ground running.
Yeah. Don't change the link of the meeting, because if you do that, sometimes the link changes and becomes
It
something
won't
else.
change.
Yeah. Okay, I see the number is still going up. Please, for those who are joining me, Pascal, are you touching your slides? Who's
Wow.
zooming them in and out?
I touched it by mistake. Don't worry. I have a touch screen. And then I forgot that's a
Oh,
touch screen.
okay. Yeah. Don't.
I was trying to take some dust off the
Yeah.
screen and
Thank you. Okay.
yeah.
I'm gonna start with the introduction because I can see that we are already past 5. We're just waiting for people to come in. Colleagues, friends, students, former colleagues, and the presenters, I would like to welcome you to our first public seminars that we hope we should be having in order to educate our people. This came about because, as you know me, I'm very actively involved with students, and then I follow them through my social media, Facebook, where we interact on various topics. And then, of course, as you know, everything these days is about coronavirus. And I thought, because of things that we are reading on Facebook and things that we're hearing, and the number of people who are losing their life, the number of people who are admitted to the hospitals, uh, one can say everything that is happening around Corona is very concerning. And as such, um, we thought it would be proper for us to at least have these seminars so that we can just try to um, uh, um, outline some of the fundamentals about this virus and the work that is currently being done, and some of the things that we can do to help ourselves to save our family members and ourselves against this virus. Because if you go out there and see the things that are happening, it's very concerning. I also want to say, on the background, we also have people who still don't believe that this virus exists. And there are those who believe the virus exists. However, they have opted to believe in some of the conspiracy theories that are making rounds. And those are very unfortunate because I can say uh, some of these conspiracy theories or theorists have managed to capture the imaginations of our people, and some of our people are believing in these things and also propagating them further. And these are leading to the demise of our people because they are unfounded. I mean, imagine, guys, someone convincing you that in near future, by just taking a liquid protein or a liquid biomolecule, you will end up being a network tower for 5G. It's very, it's laughable. It's laughable, but again, we need to respect people's views. And then I think it's a duty of people like myself as a scientist to arrange a meeting of this sort in order to explain the fundamentals that people seem to be forgetting. Because I mean, really, we are hearing things, and some of these things, unfortunately, they're coming from some of highly educated people whom you expect them to know better. But unfortunately, those are the same people who are propagating these lies. And then we can call them lies, but I hope after this meeting, we will be able to get people to understand better, so that whatever they learn from here, at least they can go back to the communities, to their immediate family members, and try to educate them, even themselves. I mean, as I've already mentioned, there are people who need education. They need to be re-educated. They need to be told the truth. And then I'm happy I managed to scoop for you people some of the best speakers in this platform. I'm going to introduce the speakers first. The first speaker that we're going to have is Professor Pascal Bessong. Professor Pascal Bessong is a virologist from the University of Venda. He is currently running an HIV research group here. He's one of the most cited scholar that we have at the University of Venda. And to the best of my knowledge, without having to suck up to him, he's the best, one of the best microbiologists in this country. So he's a very respected individual. And I expect you're going to learn a lot from him. He's going to do a very short presentation. Each speaker will have 15 minutes. And the second speaker will be Dr. Angela Harrison. I've known Dr. Angela Harrison when she was still a student at University of Johannesburg, where I was also doing my postgraduate studies. I remember, I think I was a third year practical demonstrator. I've seen her grown to become a doctor that she is today. She worked at Midtech before, where she worked on viruses. She works on HIV specifically. So she has a know-how of how viruses behave, how vaccines are developed, and all that. The third speaker was supposed to be Samie Amidou, whom I haven't seen here yet. He's going to talk about antibody therapy. And if we don't get him in, the third speaker will be, for now, Dr. Ashwell Ndhlala. Ashwell is a good friend of mine. He recently joined the University of Limpopo. He's a phytochemist.
I've been here for a long time.
Oh, thank you very much. Then I will reintroduce you, Prof Amidou. Thank you so much for making yourself available. Then Ashwell Ndhlala is a phytochemist from University of Limpopo. He recently joined. He works on plants. This guy is unique because, unlike some of us, he has 2 PhDs. And you will see from his presentation that, yeah, we are dealing with a real deal here. Some of us, we battled with 1 PhD and we never wanted to see school again, but he had strength to went for a second PhD. He's a very well-published individual. He just received his NRF rating, which is scooped the C2 NRF rating. And then I'm going to speak now about our fourth speaker, which is Amidou Samie. Amidou Samie is a professor of microbiology, also from University of Venda, also highly cited individual who has worked on different aspects of microbiology. He worked on mycology, which is study of fungi. He also worked on virology. He also did some bacteriology. Samie, I've seen Samie also. He was an inspiration to myself and other friends of mine who are also in this platform. Whereas I remember when I was an undergraduate student, Samie was doing his postgrads. I would see him in the evening carrying a macro titer plate, 96 well plate, moving around with it from his laboratory to his accommodation. So I remember him from those days, and to see him now being a world-renowned scholar is very, very inspiring. So Samie, you're welcome. I think you're going to speak third. If there's anything that you want us to do, you can maybe chat with me on the email on the background, and then we can arrange for you with Matomu. And then without wasting time, I'm going to move to the first speaker, and I would wish everyone to keep their mic off and their video off. Only the speaker should, if he chooses to have his video on and mic on, they can do so. So after the talks, probably we'll have enough time to for question and answers. If we don't, we will share with you how we're going to do it, because apparently the meeting chat was not available earlier on. I don't know whether it's available now. If it is available and you can see it, please make sure that you type your question in there, and then we can respond to it after the talks. To the first speaker, which is Professor Pascal Bessong, the floor is yours. Thank you.
Thank you, Edwin, and thank you for this brilliant initiative. I think this is something which we need to keep up going, and not only for coronavirus, but I think for so many other things as well that we are all interested in. So thank you again, and thank you for all those who have worked behind the scenes, so that we can have this, I would say, a small conversation around coronaviruses. In fact, when I was thinking of what to speak about, I said, well, perhaps I should dwell on a few things that will, in a way, add to our own perspective and understanding of what causes COVID-19. And as we were mentioning, Edwin, in terms of the background knowledge, I think it's very important because this helps us as well to have a clear idea on how to make certain informed decisions and informed consciences. So I decided to put a few slides together where to speak generally about the virology of coronaviruses, because I think if we don't have that sort of foundation, we tend to listen to other things and then we'll get a little bit confused and then we are not sure what is happening and we'll get stressed up. At the end of the day, we cannot make decisions for ourselves that will help us, our families, and the community.
To break it down as simple as possible for those of the members in the meeting that don't have a scientific background. I personally have done virus research for 11 years, and I now work frontline within the hospitals and on pandemics that occur. So I'm going to do my best to break it down as simple as possible. So the question is what is a vaccine? It's a substance that you use to stimulate the production of antibodies within your own immune system against several different diseases. Now, they are prepared from the causative agent. Now, a causative agent would be the SARS-CoV-2, and it's a synthetic or a synthetic substitute, and it is treated to act as an antigen without inducing disease. So what happens is a virus, when you get infected, it induces a disease state. This is when you become ill. And a vaccine is there to prevent that from happening. So you have to ask, what are the 4 different types of vaccines that we get? Recently, we have a new addition to this family, but the first one would be a whole inactivated virus vaccine. This is where they have completely destroyed the nucleic acid, the DNA and the RNA that's inside of the virus, so that the virus can't actively infect you. However, your body sees the virus as a whole, and it is able to form antibodies against the spike protein is the most common, but it forms antibodies against that viral. The second type of vaccines that you get is what is known as a protein subunit vaccine. This is when you are only given a certain protein of a virus. So viruses are made up of a lot of different proteins, and all those proteins working together are needed in order to cause infection and disease. So what happens with protein subunit vaccines is that you are injected basically with just a single protein like the spike protein, and the body then develops its immune response to it and builds antibodies. Now, this specific type of vaccine is considered to be one of the weaker types of vaccine, and you often require a booster or an adjuvant. Now, an adjuvant is purely a substance, an emulsion, for example, that helps your body to make a better immune response than if just the protein was circulating in your system. The third type of vaccine is a viral vector vaccine. This is when they use a vector. This could be possibly an inactivated flu virus, for example. They put the genetic sequence, the RNA for the virus, into this vector, allowing it to enter your body and your cells, producing the protein that is required, and your body then forms an immune response against it. So those 3 that I've just mentioned have typically been our traditional types of vaccines that we know of from being a baby. So all of the vaccine schedules that babies get when they are born, and as new vaccines are developed, such as the HPV vaccine, they are all of those 3 types. Now, finally, we have the entrance of a new type of vaccine, and this is a nucleic acid vaccine. This is the one that's causing a lot of controversy at the moment because it's new and people think that it has just been developed too quickly. So I'm going to give you a little bit of background on the mRNA vaccine and how it works. So an mRNA vaccine is purely a genetic sequence for the spike protein. So the spike protein is the protein that the virus uses in order to attach to our ACE2 receptors and then replicate. So what they've done is they've taken the exact genetic sequence for that spike protein and they have that injected into your system. And for this specific vaccine that we're talking about, they're using nanoparticles such as a lipid droplet to inject into the human cells. Sorry, can we please mute? Okay, so this gets into your cells and manufactures the spike protein. So your own cells then use that genetic sequence and make lots and lots of this little spike protein. But your own immune system then sees this as a foreign body. It knows that it's not normally circulating around, and this triggers the cascade of your immune system that results in antibody formation. You have your short-term antibodies that quickly come and start aiding, and then your long-term antibodies are produced. So that the next time that your body sees the spike protein, it knows how to respond immediately so that disease doesn't occur. Now, with this mRNA vaccine, there's the reason that it has come to market so quickly. There have been companies doing research for 10 to 20 years on mRNA vaccinations, and what they have been trying to formulate is what they consider a plug-and-play vaccine. This would be a vaccine model that would allow them to quickly make a new vaccination for the possibility of a pandemic that has broken out. And they do this by, once the genetic sequence of the new virus or whatever the outbreak is, they take that genetic sequence and they can then plug it into the vaccine model that they already have. This cuts down on your research. So they've already been doing that for 10 years, and they just kind of had to do it a bit quicker now because the pandemic hit. So they have been preparing. A pandemic hit, and they were forced into pushing into clinical trials to see what would happen with this kind of vaccination. And so technically it hasn't been such a quick vaccine. It's been a long-term vaccine, but when the pandemic hit and they got the sequence, it started with the clinical trials, and it was able to speed up via that process. So although a lot of people think that it's been too quick to come to market, that's the reason behind it. So what are our current options available for vaccination? Currently, there's only two vaccinations that actually have approval, and that would be the Pfizer BioNTech vaccine and the Moderna vaccine. These are both the mRNA vaccines. The Pfizer one, you require 2 shots that are 21 days apart, and the Moderna vaccine is 2 shots that are 28 days apart. The current research is showing that if you have been infected with coronavirus, that you can get coronavirus for a second time. And the booster shot is basically explaining that. So your body seems to require an additional trigger to make more long-term antibodies. So for the first shot, the efficacy is around about 50% protection. And then with that second shot, you're getting up to the 90-95% protection that your body has then manufactured those antibodies. This is not new. A lot of vaccinations require booster shots. The hepatitis B vaccine, C vaccine, for example, that's got 3 shots that you have to have, and they need to then check for seroconversion. So you go for your first hepatitis B shot. 3 months later, you go for your next one, and then there's a third one that you have to go for. And after that stage, you check for seroconversion. Seroconversion simply means that your body has manufactured those long-term antibodies. The third option is the AstraZeneca, which is not an mRNA vaccine. This is one of our more commonly found type vaccines. This is a viral vector vaccine. In the UK currently, they have approved this for emergency use, but it is still in stage 3 clinical trials. This is 2 shots that are 12 weeks apart. Then the next two are not currently being used yet. They're in the third stage of clinical trials, is the Janssen or Johnson & Johnson, which is also a viral vector vaccine. And this is going to be a single shot that they are looking for instead of having that booster shot. And then finally, the Novavax viral vector vaccine. This is in stage 3 clinical trials, which is currently running in South Africa. So there are specific hospitals that have been earmarked, and they are working in conjunction with Wits University. And they are doing the stage 3 clinical trials now. So those are sort of the first, and you must also understand there's a lot of vaccine candidates at the moment, and as they start entering stage 3 clinical trials, you'll start hearing more and more about them. So just a few questions to try and calm the waters. So the question, and there was a video that was circulating from Dr. Fauci where a gentleman asked, can I still be infected if I have had the vaccine? Since I've explained to you how a vaccine works, it's not stopping the virus from entering your body. The virus can still enter your body, but now the whole point of a vaccine is to stop disease progression. This means that you wouldn't actually become a diseased state. So the virus can enter, and because you've now had the vaccine, your immune system would then respond. It remembers seeing the spike protein, and all of your long-term antibodies come into attack. And it isolates the virus before it allows it to replicate, so much so that it would cause disease. Another question is, can the vaccine give me COVID-19? This has always been a question for people who feel that they have gotten sick after receiving a vaccine. You must remember that the whole point of a vaccination is to trigger an immune response. And an immune response happens, your temperature increases, you get some aches and pains, and you might feel a bit fluish. That's normal for an immune response. So you're not actually contracting COVID-19. Your body is just having its immune response, and you can feel safe knowing that your body has actually formulated antibodies, because there is 5% of the population who will never manufacture the antibodies even after vaccination. That's why the vaccination is only 95% effective. The question of, is the vaccine dangerous? There are risks with all pharmaceutical products, all vaccines. If you have had allergic reactions in the past to vaccines, that's when you can start considering, okay, maybe, the vaccine isn't for me. But the actual vaccine itself, that's why it has to go through clinical trials. And it has to check whether or not there is any serious concerns before the vaccine can be released. And if there is a serious concern, it is stopped. So who gets this vaccine? Pretty much anybody that is at risk. The only people that we would advise don't get a vaccine is those who have had anaphylactic shock previously with a vaccine, had a severe allergic reaction, and that would include things like hives or an extreme body rash, and this has to happen within 4 hours, right? So those type of people, we don't recommend you get a vaccine. Obviously, those that are suffering from cancer, for example, or they're undergoing chemotherapy, those type of people, that's why we're trying to vaccinate, because we need to give herd immunity to protect those people. And here's the biggest question of the day: the SA variant and vaccine options. So the current vaccines, as I explained, have been designed against the spike protein. Now, the South African variant, for the spike protein of the South African variant, there are 3 mutations on that spike protein, one of which has been identified as one of the targets that your antibodies actually form against. This means that there is a possibility that the vaccine will not be as effective against the SA variant. So what does this mean for South Africa? First of all, it's still to be determined. We don't know. They are busy with clinical trials for the SA variant to see what the efficacy is for that specific variant. And number 2, like I explained, we have this plug and play model that we have been developing for 10 years. So once they know what the South African variant is and it's been sequenced, then they can start testing the mRNA vaccine that has been adjusted for our variant, and it should be quicker to market. So even though there might be a delay or a decrease in sensitivity to the current vaccine, it doesn't mean that we just going to be left on our own and we're not going to have any options. Those options at the moment are being investigated. And that's pretty much all from me. I'll hand back over to Edwin. Thank you very much.
Thank you very much, Dr. Harrison. I just want to, before I speak of the next speaker, I just want to encourage people to keep their mic off, because sometimes we are working overtime here. Professor Amidou, if you can be loading your slides, if you have any, that would be really appreciated. But I just want to again go back to the vaccine issue to say thank you very much for giving the backgrounds and assuring us that we are on the safe side from the vaccine side of things. There has been a lot of work being done. Of course, some people are concerned that the vaccine came too early. We have been having viruses such as HIV, and why this one take very short time while HIV is still there. Those are two completely different viruses. Hopefully, after the talks, we're going to have a time for questions, and then there maybe we can be able to answer some of your questions. But now we are moving to the next speaker, which is Professor Samie, who's going to talk about another way of treating this virus, which is an antibody therapy. Some companies have already developed these antibodies to treat this virus, and Professor Samie Amidou will take us through this other way of treating the virus. Professor Amidou.
Yeah, thank you very much, Dr. Madala. Can you hear me?
Yes, we can hear you fine.
Okay, thank you very much. Yeah, so I'm going to talk about antibody therapy in the context of COVID-19. So the first question that one would ask is, what is an antibody? So antibodies, I think that Dr. Harrison has already talked about it. Antibodies are substances that are produced by our own bodies when we get infected with any other, foreign bodies. For example, mostly infectious particles like viruses, bacteria, protozoan, fungi, and many other substances as well. So these antibodies generally, when they are produced, they are specific to the pathogens that initiated their formation. And therefore, generally, these antibodies will attach to those infectious agents to neutralize them or to target them for destruction by cells. There are different cells in the body that are generally called the natural killer cells. These cells, they target any substances that are recognized by antibodies for destruction. So based on this, so there have been creation of a different types of therapy that is called antibody therapy, which uses antibodies or its parts for the treatment of different diseases. So the most type of antibody that has been used in treatment of different infections is mostly called the monoclonal antibodies. However, there are also others such as the nanobodies or human single domain antibodies that are also used. So monoclonal antibodies are full antibodies that are specifically made to recognize one specific portion of the antigen. So the first monoclonal antibody product that have been used for more than 30 years ago, and there are more than 100 such treatments that are used, and they target different treatment. So the next question that I would like to address is, have there been examples of such treatment before COVID-19? The answer is yes. So diseases, for example, such as many cancers, are treated using monoclonal antibodies, as well as many viruses. For example, herpes viruses, as well as some bacterial viruses, they are treated using monoclonal antibodies. So how does antibody therapy work? So generally, a different treatment with an antibody, one of other examples include, for example, the use of convalescent plasma. So this type of treatment is sometimes used, for example, when somebody is bitten by a snake, for example. So when somebody is injected the plasma from a recovered person, so this infuses into another patient with the disease the antibodies that are present in that plasma. In the case of monoclonal antibody therapy, this uses much more targeted approach. And we can see, for example, in the picture, how monoclonal antibody work. So generally, you have in the middle the coronavirus, And then the antibodies, they will attach to the proteins that are on the surface of the coronavirus. And then once they are attached to the coronavirus, then they make it harmless, because now this virus can no longer infect, the cells. So some advantages of the use of antibody therapy. So that particularly monoclonal antibodies, they're specifically targeting the SARS-CoV-2 virus because they originate from the blood of the people who have recovered from the COVID-19. This will potentially make them more effective than other drugs. They can be rapidly isolated and manufactured, and since the starting of the pandemic, more than 70 monoclonal antibody products have been developed against coronavirus 19. They can provide rapid protection against infection within a few weeks to some months. They have the potential to treat infected patients or to prevent infection in all individuals, including elderly, young children, immunocompromised individuals, some of whom can't receive vaccine, for example, or for whom vaccines don't always work as well. So this picture gives you different types. In A, you can have, for example, 3 different types of antibody-related products. You have, for example, the MAB, which is the monoclonal antibody, the FAB, which is a fraction of the antibody, the single domain antibody, the monoclonal antibody that targets specific domain, receptor binding domain on the virus. So you have on B what will happen, for example, on I1 in the absence of neutralizing antibodies. So in the absence of neutralizing antibodies, normally the virus, which is here in the middle, will infect the cells and then get inside the cells and cause the infection. However, in the presence of neutralizing antibodies, these neutralizing antibodies will attach to the virus, and then we can see on this side that it will be recognized by the cells, as I indicated before, for destruction. So this is also the mechanism of action of monoclonal antibodies, how they work. So are there currently antibody therapies developed and accredited for the treatment of COVID-19? Yes, indeed. As Dr. Madala has indicated, the FDA has already given emergency use authorization. So remember, this is different from registration, the regular process of recognizing the use of a specific drug in any country. So this is emergency use authorization, and this is because they have weighed the negative effect against the positive effect, and they saw that there were more positive effect compared to the negative impact. So the FDA has given emergency use authorization to 2 monoclonal antibodies treatment for COVID-19, and these include particularly the bamlanivimab product that is produced by the Eli Lilly company, and a cocktail or of two different monoclonal antibodies, that is casirivimab and imdevimab, developed by the Regeneron. So there is another group, another that is now on phase 3 trial by AstraZeneca, which is also a combination or a cocktail of 2 different monoclonal antibodies. So I would like to indicate that the Regeneron product combination was the one that was used to treat President Donald Trump when he contracted COVID-19 in October recently. I think that everybody will remember that. These molecules, they target the spike protein. You heard Prof Bessong talking about the spike protein, and Dr. Harrison as well. These are the most important protein that participate in the in the infection, in the entry point of the virus. So the spike protein of the virus and stop it from attaching and entering the human cell. So when these antibodies recognize the spike protein, the virus become incapable of getting inside the cells. So the shortcomings of the antibody treatment, because we talked about the advantages, but there are some disadvantages. One of the main shortcomings of the antibody treatment is the high price, because remember, it is a very complicated process. The production of monoclonal antibodies is quite a complicated process that it makes it quite a bit expensive to produce. And you'll see that most of the monoclonal antibodies produced that are mostly used in developed countries because of the price. I think that one of the cheapest monoclonal antibodies that have been produced is from India, because they have their own companies that can do that. So also we have limited supply. So the product by the Lilly company, as well as that of the Regeneron, they were produced in the United States, and most of their product was already has already been bought by the American government for their own people, and most of these products have already been used. So there is limited supply, and they might not be able, to supply throughout the world, except if each and every country was able, to produce their own. So we, scientists in South Africa, we need to make more effort, and particularly appeal to the government, to encourage, such research. I would like to particularly, appreciate Prof Bessong and his colleagues, who are involved in the COVID-19 research, because we really need such. There are many people who have, ideas, but they cannot implement because of the lack of, research, and, the government lack of interest also, to encourage such. So the other limitation also is that it cannot be given to people who are already hospitalized. So basically, if somebody is already hospitalized or is already in a state where they need oxygen, so then they cannot be given, the antibody. So the antibody will actually be harmful for them. So they can't be given, the antibody. And also, I think one of the other things that for now, it cannot be given to people who are less than 12 years old. So, and then, of course, they are still in experimentation and many others to come. So in conclusion, I think that antibody treatment is still in experimentation, and any government should take responsibility in developing policies around their use in the, in the country. You can see, for example, the case of the US, the different monoclonal antibodies that have been given emergency use authorization while waiting, the confirmation of the, final clinical trials. I'd like to thank you very much, and indicate that data that I've presented here were published between November 19 and December 21, 2020. Thank you so much.
Thank you very much, Professor Samie. Dr. Ndhlala, if you can be loading your slide. I would like to just make a few comments from your slides that indeed it goes to show that it's not only vaccine, people. So if you think people want to inject you with something that you don't want, there's still antibody therapy. There are many, many ways. As we will see now with the next speaker is going to give us another type of treatment. This will be our last speaker, Dr. Ndhlala. He's going to talk about phytomedicine, phytotherapy as an alternative means of treatment. I know everybody from their houses, people are steaming, they are using different plants. We can see now that in Venda, mibomo iko fera au chana mibomo zvumbumbe zviko fera vatu vaku steamer vaku aravera kachivenda. So we really want to understand whether if indeed this steaming works, because people they pretend that they're not doing anything, but inside their houses we can see steams coming under the doors. We can see the windows are covered with steam. We wonder what is happening inside. So and people are using different plants altogether. But there are certain plants that works towards this. Some plants are very, very toxic. The plants that when you steam using them, sometimes you can turn blind. There are plants which when you use them, you can damage your kidneys. There's plants that are very cancerous if you inhale some of the compounds that come from there. So we want to help you people, because we know some of you are ashamed of steaming. So when you do it even in private, at least do it with the correct plant. So Dr. Ndhlala is an expert in plants, so do I. I'm also an expert in plants chemistry. We need to help you guys so that we don't do it. I wonder people from other cities like Johannesburg, Pretoria, and Cape Town and Durban, what type of plants you are using. So Dr. Ndhlala is not Venda, so he's not going to use the plants that are found in Venda, Bomo. No, he's going to give an overall or a general plants that we can use. Sometimes maybe you find that we can even use grass, and the next thing you are there on top of the eucalyptus tree trying to get the leaves, while you can just use grass. Alternative medicine, phytomedicine, herbal medicine as an alternative medicine for treatment of coronavirus. Thank you very much, Dr. Ndhlala.
Thank you, Edwin. I hope I'm audible, and I hope my slides are showing.
Yes, you are audible. The slides are showing indeed.
Okay, thank you. And good evening to everybody. Edwin asked me to talk about the role of herbal medicines in this era of COVID-19. In general, it is the research that we are doing, which entails how can we use plants, or how can you use resources around us that are in other words nature given, or in some other words God given, to see how we can combat whatever that we encounter as we take this journey of life as living beings. And our research is mainly stemmed from a long time ago. We thought we would need to go back into the journey of life and understand exactly how do we come about to know plants can be used as medicines. And we all know that the earth was actually formed about 4.5 billion years ago, after a very violent bombardment with stones from the universe, as well as burning gases within the earth itself. But what really perplexes us as scientists is life still emerged from this. So what does that mean? It really means that within our systems, we have got ways of fighting something that is coming against us. So we have got ways of fighting our own enemies. And those ways that we have developed, some of what we have heard earlier in the talks before, the antibodies, the use of vaccines, and the use of other pharmaceuticals. But there is another way that we can use, which is what I mentioned before, which is nature given. That is the plants. And what happened after that bombardment that happened about 4.5 billion years ago? What it did give birth to was several mega diversity or hotspots, which in science we call floristic kingdoms. And one such kingdom that we have is the amount of plants that we have in the Southern Africa that we call the Capensis kingdom. And from there, as our forefathers learned how to use these plants, there was trade all over the world, exchanging these plants and exchanging knowledge in terms of how these plants can be utilized. So this is one of the terms that I like to use when I describe the immense wealth in terms of plants that we have. So I usually call it a botanical gold mine. So we have got about 15 centers of endemism in Southern Africa. And this gives us vast opportunities in terms of development of medicines, in terms of development of other industrial chemicals or other agrochemicals that are used not only in fighting diseases, but in other applications as we sought to live as humans. So this actually, as years progressed, we have learned how to use this, and one form of a system is actually been born from there. And that form of system is called traditional medicines. So this is when we learn as traditional people, or as people, as communities that stay in one area, like what Edwin mentioned before, that I may not talk about Venda plants, I may not talk about Johannesburg plants. It's because each community, as they sit in one, they identify specific plants that they use, and they identify specific parts of those plants that they use. So you may find that one plant can be used as leaves in Venda, and when you go to another province, they use the roots. But in all, we call that integral system traditional medicine. And our forefathers, and not just our forefathers, but many of people who have got that knowledge, has been passing it from generation to generation. And now, as we do our research, one thing that we aim to do is now to document those uses. And COVID-19 is not an exception in this. So we are busy now doing vast work in terms of understanding and in terms of researching those that our indigenous people are using. So one of the plants that they use is the Artemisia afra. They also use Lipia, Zumbani, if I speak in another ethnic language. But all in all, what we need to do as a nation is to come together as scientists and understand how these plants work, and then understand that these plants also can then get depleted in the wild. Because if you see, there's one picture that I've put here, somebody has used a whole bunch of it. And if everybody's doing that, then at the end of the day, the demand will get higher and those plants will also go extinct in the wild. But now, coming to the question which everybody may want to understand is, do these things work? And I want to tell you, yes, they do work in many different forms. And if you're talking about respiratory diseases like the pandemic that we are facing, one of the main intricate way of doing it is steaming. And a lot of people has now come up to light in terms of doing that. But there are also some that can be used as herbal teas. There are some that can be used as drinkable medicines or as prepared medicines. As you can see that in South Africa, the history of how to make these mixtures is come a long way, and we have perfected this art. And in many ways, it has helped in many occasions. But we also now to give a bit of credence in terms of what these plants can do. So what we do in our laboratories or in our comfort zones when we do our work is to take those plants and go out there, talk to the communities, and understand how they use those plants. And then we'll take them to the laboratory, and we have developed what we call models that we can test. For example, if we know that we are dealing with a virus, we have got models that we test within the laboratory that can show us how much that plant can actually inactivate a virus, or how much that plant can actually assist in terms of reducing the symptoms of what that virus is causing. And obviously, we do it if we want to take our medicines into mainstream medicine, and that is what we are aiming for, to take this whole traditional medicine into a whole new other direction, that we can bring it into mainstream medicine. So we have got many programs that we are working with, students that are working on different aspects in terms of this virus that we are dealing with. But just a few facts we now know, and we have been told even in the previous lectures, that the main culprit is the SARS-CoV-2. That is the main culprit of this pandemic that we are living in, and that is the agent that is causing COVID-19. And as of the current statistics that we are looking at, it's about 97 million that has been affected worldwide. And of those, about 2 million has already lost their lives. And if we are narrowing it down to South Africa only, it's about 1.3 million infections, and about 40,000 have actually succumbed to the virus. So it's just a serious matter, and as scientists, we need to go out there and bring whatever that we can to try and save whatever we can save. And as we all know, and as we have heard, that even the vaccines itself, there is still some information that we still need to learn as scientists. So there is no safe and effective antiviral that has been developed so far to treat the pandemic or to treat the disease. And there are no pharmaceutical interventions either. Even though we have got vast in our cupboards or in our hospital lockup that can be used in terms of reducing this load. And most of these, you've got the background from the plants. Some are actually synthetic. And we also have heard, I don't want to go back into that this virus, as we know it, it's RNA-based. So it works exactly or more similar to the HIV, because what does it, when it affects, then needs to change back from RNA to DNA, and then the rest can occur. So as plant scientists, we need to understand that the way that the virus look, so that when we use plants or when we test the plants that we are given, we can then identify what we call mechanisms of action. Where do they target and how do they do it? And one of the things that we have seen is that the weakest point of the virus is its external protein structure, which shows those spikes that we see when they depict the virus to us. And those spikes are the ones that actually enables the virus to enter easily into the human cell. So those spikes, they actually facilitate the infection of human cells by the virus. And so what we now probably need to do is probably identify therapeutic agents from plants that target those proteins, or that targets the way that the virus attaches itself using those proteins, and then we can then come up with plants that we can recommend. And as we can see now, it's a battle against time, and we cannot say we are losing the battle, but the virus is also on the other way, battling against us. And we all know now there are so many variants that have emerged. So far, we public knows about 2 variants, the SA variant and the UK variant, apart from the main initial SARS-CoV-2 that we started with. So these other 2 variants, they cause more havoc. And as we believe it and as we understand it as humans, it's not only those 2, there are many other variants that are probably still hidden somehow that will come out. So we need actually to be very, very vigilant in terms of what we use and in terms of what we do. So I think this has all been talked about before, but only for me, I'll be interested in, because most of these plants, they produce a group of compounds that we call polyphenols. And these polyphenols, as we now understand, can actually play a part. This will be the human cell, and this is the virus, can actually play a part by blocking where the SARS can attach to before it gets into the human. So that's how most of the plants, if they are used as drinkable herbs, can work. But we all know also that there are some plants that we know that produces oils. For example, one of the oils that has been largely used today here is the essential oils of eucalyptus. So we know that they go and disrupt the membrane system of the virus, and then it disintegrates, and after that the virus will be inactive. And also looking further into the other creative way that we can use with plants is when the virus gets into the human system, and like I said before, it has got RNA as its gene mechanism. It needs this RNA to be changed to DNA before it can produce more of the virus itself. So if we target any stage within this cycle, then we can win. And most of these natural compounds, this first stage here is what I've just described, that these compounds can prevent the virus from entering the human cell at all. But okay, when this doesn't work, when the virus gets into the system, what else can we do? We then have got compounds that can increase our immune system by boosting the immune system, or we have got compounds that can actually prevent any one of these stages from occurring. Either enzymes that are involved in changing the RNA into DNA, those can actually be blocked, and then we don't have further production of more viral particles within the system, so that then we win against the war against COVID. So this is also just another illustration as to how plants can be used. If the virus gets in, then we have to target any of these compounds. So this is just more or less like a similar illustration from before. But now going to specific plants that we have done simple experiments with. And this also goes, maybe if I may take this opportunity to demystify some of the things that has been going on, I will probably bring more slides that will demystify other things. But here I want to specifically talk about, there was a video that was going around that had a lady from the WHO who was talking about South Africa growing the virus. They said it in a very, very layman way to say South Africa is growing the virus, and most people took it in another way. But in actual fact, what we are doing is to use the virus in the experiments. So that's the only way we can know whether it works or it doesn't work. So what we do is we grow the virus in specific medium, and then we keep it in nitrogen or we keep it in any other form that doesn't allow it to be exposed to other people. So then we take our plants and then we do our extracts and then we test it against those virus or try to get any enzymes or any stage that we can inhibit if we know specific enzymes that are involved within any system as it gets itself manifested into the human body. We then use those enzymes and then we took them and then we put them in a laboratory setup and then you inhibit them against their functions, and then that's the only way you can know that the plants are working or they are not working. But here I've just a few plants that I have. Artemisia, there are two types of them. There's one that has got compound that were used mainly to cure malaria, but recently it has been used more often in reducing the effects of COVID-19. And information that we got from our forefathers that a long time ago, they used to burn the leaves of Artemisia. This representation is not Artemisia, it's in paper, but they would smoke it like this, and then they will inhale the smoke, and the smoke will equally cure or equally clear off the nostril away and relieve anybody of any strong flu. So that is exactly what the transition came from, and it seems to be working when people use it for that. And then we have got eucalyptus oil. It doesn't really cure, but what it does, it assists with unblocking the congested nostril. It assists in clearing the airway. And we have got moringa. Moringa it's a powerhouse in terms of nutrients. It boosts the immune system, and it has got all sorts of polyphenols that we know can actually probably bind and reduce the rate at which the virus can infect a human being. So we've got many types of mechanisms that we have seen that can occur when we use the plants. We have got what we call the S2 blockers. So the S2 blockers, we know that the S2 is that receptors that allows the coronavirus to enter into the cells. So if you have got plants that produces caffeine, it's a compound that is mainly found in coffee and many other green teas or the black teas. So what it does is it binds onto the receptor that also bind to COVID. So then there is competition between the COVID and these compounds in binding onto that receptor. So you reduce the rate at which you get infected, or you reduce the chances of you getting infected. Then we have got the natural product that targets the TMPS2. This is an enzyme that when the COVID binds to that S2 receptor, and then it gets itself into the human cells, there's need of an enzyme to reverse that binding. So this is what happens here. So we know that if we inhibit this enzyme, then there is no that reverse doesn't happen. So then you reduce that chance of also getting yourself infected. And we all have the anti-PPRO active plants. This also like more similar to the HIV mechanism, where the genome is in the form of RNA, and then it needs to be translated into DNA before then the virus can produce more of its own copies. So now if we target those enzymes, then we block and we reduce the rate at which the infection occurs. So besides that, we have been advocating in planting and making vast pieces of land available in terms of planting those plants that we think can work towards that. And some of the work that we've been doing, we've been merging what we call indigenous knowledge system with science and technology. And we've got very knowledgeable students who are working into even using technology computers to try and predict where we can plant those around South Africa, and hopefully we are going to expand into Africa. And then we see that we have enough raw material in terms of what we plant. And also, this was work that we also did with Edwin. And these compounds are very key in boosting the immune system as antioxidants and clearing the immune system, which is also key in fighting any disease. And COVID-19 is not an exception. We have got oils from moringa also that we have seen that they contain specific characters that can also work synergistically with other compounds or other medicines that you'll be working with in terms of enhancing or being vehicles in getting those pharmaceuticals to their target points where they are supposed to act. And then we have been in a drive to make products out of some of these plants and out of some of these compounds. And these are just a few that we have made with moringa iced tea. There's vitamin water and many other forms that people can palate. And this will probably be in more or less a preventative stage where somebody is not infected, but they want to prevent infection. But then we have got tablets and capsules and powders that can be now used by people who are in severe symptom of the disease itself. But as we use plants, we know that there are some myths that are or specific things that we know really, like what Edwin mentioned before. There are issues of toxicity in plants. We don't know what happens in long-term use, even though these plants have been used for thousands and thousands of years. But also in this era, we know that people are trying to make money. If you drive from Johannesburg to Polokwane, for example, the whole road is full of people who are holding mshonyane, which is artemisia, in the road selling to people. So these people, when mshonyane runs out, they still need money. They just go grab another plant and then they sell it to somebody who doesn't know what artemisia looks like. So those are some of the things that we need to deal with, and we need to try and stamp down. And also the issues of contamination, because you don't know how those people keep those plants. Do they keep them under their beds, or do they really keep them in a safe, cool place like what we encourage? How are they hygienically keeping those mixtures that they are selling? So it's something that we need to stamp down in as much as plants work, but these are very big dangers that we really face. So in doing so, we are coming up and we are working very hard and tirelessly, not just in South Africa, but we are also seeing African Traditional Medicine Committee to come up with standards that deal with the ingredients of those herbal medicines. How do you harvest those things? What the microbial limits should be, and what are the test methods that we should use when we are in the lab to see that this really works against the virus, the calculations, packaging requirements. So all this, we can then now take them towards working together with the vaccine or trying to complement what the vaccine is doing. But then we know that there is now a lot of things that has been going on around internet, around social media. Some of them has already been dealt with here about how unsafe the COVID-19 vaccine in terms of its development. Why was it so quick? And this has already been answered that yes, it has been produced in a record time. Why? Because we are no longer in the 80s, we are no longer in the 70s, we are now living in the 2020s. So the technology that we have in the laboratories is awesome. We can produce things that we could produce in a month. Now we can do it in a day, we can do it in an hour, we can do it in a very few space of time. So that cannot be something that can make people draw back. Another thing is, can I get COVID-19 from the vaccine? It has been answered. The vaccine doesn't have any life in it, or it doesn't have any virulence in it. So it's just a shell that is meant to mimic what the virus is like. Will I get the DNA? You won't get the DNA because it only comes in RNA. There's the side effects. It's like also has been said that everything that is pharmaceutical can produce a bit of side effect, but they are not fair compared to what you then get. Vaccine causes infertility in women. Of course, there is a part of the virus, the spike in the virus has got a part of it that has got the DNA strength that is similar a bit to a placenta cell, but it has been shown by scientists that that is not at all going to be an effect. So just to quickly finish my talk, I probably have been talking for long, but there are also other mysteries that has been going around. For example, this has been circulating on the internet, and it has been seen here that it's written not for US and Canada, but this, first of all, it's not a vaccine. It's a medicine that is used in that, or a generic medicine that has been produced in India, and it's used in many other things, even though it can also assist in COVID-19. But actually, this is not a vaccine, and this is because of the licensing, so that it cannot be sold in the US and Canada or EU on the black market. And obviously, just to conclude, medicinal plants and natural products are promising, and we need to work hard towards development of these, even though there are politics involved, emotions, and there's need of money to do the research. So that's what we need from our government. I thank you.
Thank you very much, Ashwell, and thank you very much for people for being patient. For to have 4 speakers, it was a big task, and indeed we were all here to listen to this, and I thank you. But we are going to take only 5 questions for now. What we're gonna do is that we're gonna ask you to leave your questions. We're going to leave you a platform. We're going to share the presentation. Maybe next week we can have another session, if possible, to get some of our speakers to answer to some of your questions. I can already see 2 hands that I'm going to take immediately. I'm going to unmute Professor Piata, Lizelle Piata, who's the head of the Department of Biochemistry at the University of Johannesburg. The floor is yours. If you can unmute your mic and give your question.
Thank you, Dr. Madala. I just want to start off by saying, number one, thank you for organizing this talk, because it's most wonderful to have such 4 speakers whose names you've heard of before, but you're not quite sure of what they are doing. And to imagine that you can use them as guest lecturers in for example, honors courses. For one, Dr. Harrison will remember doing vaccines actually with me. And I hope she won't be offended if I actually do contact her for a further talk. Having said that, I have 2 questions. The first of those is, are any of the panel familiar with the mutability of the specific amino acids in the South African versus the UK variant? Because as far as I know, it's an asparagine that's mutated to a tyrosine in both of the strains, except our strain has a few more mutations. That's my first question. My second question is, and I'm very happy that the South African colleagues are alluding to alternatives than vaccines, because according to my knowledge, a lot of money is being pumped into only the vaccines when drugs, or as Professor Ndhlala alluded to, inhibitors of specific things are being overlooked. So I'm glad to see the alternatives, and I'm wondering if anyone knows or is aware why so much money is being pumped into vaccines which are mutable, at least the strains are mutable, and not drugs, and drugs which are inhibitory. And I also just want to make a comment to Professor Ndhlala that since you have this wonderful biodiversity, do please protect it so that the Americans do not do what they've done before with the hoodia. I thank you all.
Thank you very much, Dr. Piata. So I hope the speakers are noting the questions. I'm going to move to the second question, which is going to be by Professor Safi Traore, who's the head of the Department of Microbiology at the University of Venda. Safi, the floor is yours.
Yes, thank you very much, Edwin. Thanks for the speakers. It was really an eye-opener, and really we appreciate that. And thanks for inviting us. I just want to comment on the variant. I was just watching CNN now, I don't know, BBC, and they just discovered today a new variant in the Amazon. And apparently it's like a combination of the South African and the UK one. So I don't have a question. Just this was my contribution. Thank you.
Thank you, Professor Traore. I think that will also add into the question that was asked by Professor Piata with regard to the changes that are happening and versus the vaccine. That will be also answered. Katu, if you can unmute yourself and ask a question, please. Katu, are you able to do that?
Yeah, can you hear me?
Yeah, I can hear you very well.
Yeah, thank you very much. My name is Ashwell Ndhlala. I'm from South Africa, Limpopo. Right now I'm in Germany. The thing is, I didn't really hear so much of the side effects of vaccination because I heard that somewhere in one of the Scandinavian countries there was a problem of people having developed sleeping disease after they've been vaccinated. You know, you remember many years ago there was this outbreak of swine flu, and then people got vaccinated, and now I think it was Sweden, if I'm not mistaken, they're not going to vaccine anyone anymore because of that problem. What can you say about this as we are talking about the vaccination at the moment? I think it's very interesting to also get to know what could be the side effects that people should be aware of as we are heading towards, vaccinating people in South Africa. Thank you.
Now I'm going to take, okay, maybe before I take this second set of questions, let's give it to our speakers to answer. I'll first give a floor to Dr. Harrison, who spoke mainly about the vaccine, and then maybe Professor Amidou will also answer, give her his own perspective, and then the other speakers will deal with the second set of questions. Dr. Harrison.
Okay, so thank you very much for the questions. Hi, Lizelle. Yes, you're more than welcome to contact me afterwards. Okay, so number one question is, why is there so much emphasis being put on vaccine as opposed to treatment? So while the vaccines have been being developed, there is a lot of research going on with the possibility of current treatments. So a lot of my background has been in drug development for HIV drugs. And basically what you have is drug platforms that they try and repurpose current drugs because you can get quicker to market with them, because they've already gone through the clinical trials of, you know, the side effects, that kind of thing, and they have proven safety in the human population. Basically what's been happening is, so far the current treatments that they have been finding, the result is a lot longer to get to than what they've been able to do with the vaccination. And what they're doing with vaccines is it allows you to have a mass treatment of a lot of people, all in one go or in quick succession, to create that herd immunity. While this is happening, they're still trying to find certain drugs that will counter the virus. And like has been said, there is still quite a few, and there is natural medicine that also helps. This is a toss-up. It's basically a pandemic, and as soon as we can get a solution, that's what's being pushed out. And the first solution that came to light has been the vaccinations, because they've kind of been preparing for a possible pandemic for quite a few years. With regards to the mutations in the SA variant as opposed to the UK variant, first of all, I think it's important for everybody to understand that when they're labeled as UK and South African variants, this doesn't mean that the mutation happened here. It shows the UK and South Africa have one of the biggest monitoring systems in place for genetic sequencing. And so we do that monitoring and surveillance in this country, and the UK does it as well. The chances are this mutation came from America, for example, and it just happens to be that we discovered our variant. We discovered the variant circulating within South Africa, and then it is reported as a South African variant. So the mutation that is common between the South African variant and the UK variant is the N501Y, which is where the asparagine has been converted to a tyrosine. These are all in the receptor binding domain. So all three mutations that have occurred for the South African variant occur in the receptor binding domain or the receptor binding motif. Then the second mutation that has occurred within the receptor binding motif is K417N, and this is where lysine has been changed to asparagine. And the third one is E484K, where glutamic acid has been changed to lysine. And the effects of this for us for vaccine, as I explained before, they've found that the vaccine is still effective against the N501Y, but now they're still doing research to check whether or not the addition of the two extra variant changes is still going to be affected by the vaccine. And unfortunately, it looks like there's a bigger possibility that it won't be as effective, simply because there is so many of those mutations. Having said that, the N501Y mutation, this is actually one of the when they've tested the antibodies of patients who have seroconverted, this is one of the targets of the antibodies, and that indicates that even the vaccine won't be as effective against the UK variant either. I hope that answers those questions. And then as far as the questions were concerning about the side effects of vaccines, pharmaceutical-wise, vaccine-wise, this has gone to market quickly, as we say, and the clinical trials are going pretty quickly. With any drug that is researched, with any vaccine that is researched, they have a scale of what they considered an acceptable side effect. So, for example, the side effect may be a headache. Now, for a cancer patient, that's not considered something that you would be worried about, because cancer is more severe than having a headache. But let's say, for example, a headache was the result of a stomach medication, then they may think more about, changing something up there and it not being an acceptable side effect. With more and more of the vaccine rollout, you will find more and more people reacting differently. And this goes across race groups. It goes because everybody has their own. This is why they try as much as they can when they do clinical trials to capture as many of the different populations as possible, because everybody can react slightly differently. It always helps to be able to include all races in the vaccine trials. But as it starts going out into more remote areas where it isn't so common, you will find more and more listings happening of various kind of side effects that are happening. And against, they have to weigh it. If it's causing deaths, then they have to investigate whether or not that death was directly related to the vaccination. If the people had a side effect to any of the excipients that were in that vaccination. Excipients are items that are added to make it liquid, for example, or in a tablet to bulk it out because it's such a small milligram dose. There is tons of different reasons, and what they do with these clinical trials, and once it's rolling out to the human population, they list every single one of these reported adverse effects, and they go and they start investigating one by one. But it's still weighed up on population percentages and statistics as to how many people had a possible death as opposed to how many people didn't. And there is a an acceptable threshold of negative and adverse reactions that is allowed under circumstances such as a pandemic, where we are at risk of losing millions of people as opposed to 100 people, for example. From my side, I hope that covers the questions.
Thank you, Dr. Harrison. I think we are against time. Professor Amidou, is there anything you want to add?
Yeah, hello, not really. I think that Dr. Harrison has addressed all the questions. One thing that I just wanted to say about the intervention of Professor Pieter was to say that, in fact, this is an important opportunity, to get in contact with colleagues, and that gives us an opportunity to get to know what other colleagues are doing, and probably initiate some collaboration. And I think that this is a very good platform to start such collaboration. I'd like to again thank you, Dr. Madala, for such an opportunity, and I hope we are going to take it from here and probably build up some collaborations and some friendships, that could lead to probably better, contribution to science. Thank you.
Thank you. Now we're moving to second set of questions. I've noted Dipuo Makubela. I've noted Edmo Shonai. Dipuo Makubela can go first, please.
Oh, hi, my name is Dipuo. I'm a clinical trial assistant at Merck Research, and we are currently working on 2 vaccines that Dr. Harrison have mentioned, that is Novavax and Janssen. So far we are administering the vaccine. We're not administering the vaccine to pregnant women, or we are encouraging people to be on contraceptives. I stand to be corrected, but I believe this is because studies have not been done on how the vaccine can affect the pregnancy as time goes on. So my question is, could there be a possibility that it might in the future affect the gestation period of the pregnancy? That is for participants that have already been administered with the vaccine. Thank you.
Thank you, Dipuo. Professor Edmo Shonai.
Thank you, Dr. Madala, for organizing the meeting, and to the presenters. We really appreciate what they've shared with us. I just have a question regarding the vaccine. So we know now, or at least there are reports coming out, that if you get infected, you could be protected for about 6 months post-infection. And given the pace at which the vaccine has been launched, I mean, it's record time, what assurance do we have with respect to long-term immunity? I speak here like a lay person with respect to immunology. And I also don't know if it's enough to explain the rate at which the vaccine has been launched, that there's been a cassette waiting, you plug in and there you go to the market, because I think this builds into the conspiracies, around, the rate at which this has come about, and the pre-planning. Maybe science has become a victim of itself, of its own efficiency with respect to predicting there will be a pandemic, and then we have a cassette ready. I think that that in itself, you can see build some kind of conspiracy theories. I don't want to speak like I'm one of that, but I think we need to be open about what the community thinks when they hear about this. Then I have to address one more issue with respect to the herbal medicine. Being an African, I think I must acknowledge that we are underestimated. I remember Madagascar coming out, well, maybe not so much in a scientific way, but in a traditional sense, talking about how they manage respiratory diseases and that they were going to do this for themselves. And we know the backlash they received. And if we look at Africa now, in spite of our poor health system, I think we've been holding out fairly well, maybe on account of some of these interventions. And I think it's time Africa came out and defended their own approach to some of these things. Thank you.
Professor Shonai, quickly, if we can move to Gavi. Gavi, are you still here? If Gavi is no longer here, I will give the last question to
Dr. Madala?
I think the network is a problem.
Okay, the network is a problem. At the meantime, because we can't get them to answer, we can wait for the questions. Professor Pascal Bessong, are you still here? Professor Bessong, if questions, please, I mean, in 2 minutes. Dr. Harrison?
Hi, yes. Okay, so basically, as far as the pregnancy concern and making sure that all of them are taking contraceptives is like you said, they haven't done any clinical trials at the moment with pregnant ladies. That meaning that we can't administer to pregnant ladies, and anybody who is receiving the vaccine needs to be on contraceptives, because at the moment it's we don't know. It's still to be determined, because the virus is a new virus and it's a new pandemic. There is a lot that is still to be determined. Then when we're having a look at the system as far as science, it is a victim of itself. We have to predict what's going to happen, unfortunately, and just with drugs, we try and predict, and we try and see where those mutations are going to happen ahead of time. And so, yes, that does feed into the whole conspiracy theory of pre-planning that does. And then the last thing I'm just going to address is the reason why Africa has such low infection rates and the death rate is much lower. It is often very underestimated just how robust the African immune system is. Number one, we are always in contact with a lot more infectious and a lot more bacteria and a lot more fungi. We tend to be outside more often and in contact with all of those things. And from being a baby, your immune system is much more robust than what is being seen in the European sector and America, for example. So definitely, I think we have handled it a lot better than most of them. And again, I reiterate that the immune system for the African continent is far more robust.
Thank you. Dr. Ndhlala, just a comment with regard to the phytomedicine quickly, please, 2 minutes.
Yes, I agree with both comments that we really need to come out and defend ourselves, but we also need to bring out that scientific evidence. Not that we saying we are not trusting ourselves, but I think those are the systems that has been laid down, that we need to do clinical trials. We need to do all these things for it to be put into mainstream medicine. Thank you.
Pascal Bessong, please talk about the issue concerning a person who has been infected by the actual virus can only have an immunity that will last 6 months. But if you take a vaccine, you're going to be protected for a lifetime. Is that a myth or a fact?
Well, you know, once a vaccine is made, you know, one of the things. Well, let me take a step back. You know, we have been receiving vaccines all our lives, and we have vaccines which we know can protect us for 6 months, for 1 year, for 5 years, and even up to 10 years. And those are all established things. And we also know that, and some of us also develop a natural immunity. For example, against hepatitis B virus. And many people, in fact, almost 80% of us pick up that virus when we are babies. But a few of us will develop liver cirrhosis at an advanced age. And it's been shown that most of us actually had a natural protection. So in that case, we may not need a vaccine. But as Samie mentioned earlier on, you had some vaccines which and you are given antibodies produced by another host injected into you. Now you are not the one producing those I would say antibodies, and they don't last over time and they finish. And so, for example, anti-tetanus vaccine has a very short lifespan, and the vaccine for cholera is about 6 months. So you see, there's a variety, in terms of protection, artificial as well as and the natural. So I hope that answered that question. But I want to touch quickly on the aspect on the prediction of disease outbreaks. In fact, humankind has always reacted to outbreaks. And one of the ways, as a way of preventing, to get ourselves ready, because we will always be exposed to infectious agents. It will never, ever go away until the kingdom come. We can, and we don't know, there are a variety, there are millions of pathogens, I would say potential pathogens in our environment. And with time, they get used to infecting us and they're going to cause outbreaks. So one of the ways to try to mitigate that is to try to have an early warning system. And that's the prediction we're talking about here. That if it does happen, at least we have some things to respond to, even if we don't respond as much as we expected, but we will try to reduce the harm that may cause human populations. But I agree with Shonai that perhaps the way that is packaged may give the impression that we already know. We don't know, but we want to have a clue of what may happen, and such that we can prepare in that regard as much as possible. Then the last point is that, and data came out last night that shows, and that the mortality from COVID-19 in Africa is higher than the world average. So data is beginning to come out. And I think we need to be very careful when we say that the African populations are actually resistant to COVID-19. Most African countries don't even collect data enough that is quite reliable. We don't know how many people in Africa have COVID-19 or are infected in the first place. A few countries, and South Africa is one of them, that has a national collating center and gives you a certain level of credibility to what is coming out. Even then, it is not entirely complete because many people have not tested, and some people, if they are sick, don't go for testing. So what we see, for me, I think, is the tip of the iceberg. But as the gates are opening and more info is coming out, we will start having a real glimpse of the weight that is in our hands. And that is why the prevention, and somebody asked the other time, why do we spend so much money in terms of the vaccines? Is because there is a strong optimism that the vaccines perhaps may give us a window of a chance and to reduce as much deaths as possible before we actually get and some curative medicines. Again, let us not forget that there are very few viruses that are actually cured by drugs. Very, very few. Most viral infections have been eradicated or controlled to an appreciable degree through vaccines. Over to you. Thanks.
Thank you. Thank you, Pascal. I'm just going to run through because we're left with only 5 minutes. Professor Reinhold Mayboom, please, I'm giving you only a minute to ask your question.
Thank you, Edwin, for the actually nice initiative. I'm trying to keep this within a minute. I've got two more or less philosophical questions for the panel. The first question is, humankind has been active in agriculture for about 5,000 years. And in that period, they've been using guano, in other words, bird shit and bat shit, as fertilizer. So in other words, there's been interaction with bats for the last 5,000 years. So the first question is, why is the media, and this is a philosophical question, why is the media presenting this as a new situation if the interaction was there for several millennia? Knowing that in 2001 it was warned that there might be a coronavirus jumping from these specific bats to humans. Second question for the panel is a very simple one. There is a lot of media attention on a vaccine, but absolutely none about repurposing old medicines. And this has been partly answered already, but I just want to bring it to attention again. Dr. Hill gave a fantastic webinar for the South African public in this week about showing how ivermectin, for example, was possibly a good agent to fight COVID-19. There might be different philosophies whatsoever about what the causative effect is, but there has been absolutely nothing in the media or in the funding agents about repurposing medicine to see if it works for COVID-19 and increases the survivability of patients. So those are basically my two thoughts, philosophical questions. Thank you. Edwin, I think I kept it under a minute.
Yes, you did. Gavi, we've been trying to give Gavi a chance to ask the question, but Gavi is not here. Gavi, can you unmute and ask your question, please?
Can you hear me?
Yeah, we can hear you. Please quickly ask your question.
I just wanted to squeeze in a question as a politician. I'm not from a scientific background, but I wanted to ask the panel. From my understanding, we've seen prominent people taking the vaccine, but yet we're still seeing people saying stuff about the vaccine, saying that it's not legit and all of that. Shouldn't that be a proof enough that the vaccine is legit? If you see people like Rupert, the Queen, and all these prominent people taking the vaccine. Thank you.
Thank you so much. Professor Amidou Samie, please answer Gavi first about the issue of prominent people taking the vaccine. Why is that not enough to convince us that the vaccine is not a scam?
I think that there are several issues, that are at play. As Professor Shanai has indicated, and also the professor just talked right now, there are certain behavior of particularly of the media that make people, suspicious. And once people, have become suspicious, it's very difficult to convince them. Of course, we can talk about the science of the vaccine. We know how the vaccine works, what is in the vaccine. But when people start to look, to see certain pattern, certain behavior, for example, the question as to why is it that only the vaccine is being pushed forward? Of course, Professor Bessong has, given some explanation to that the vaccine gives us a much better chance, of preventing the disease. And you can see that most viral diseases have been, eradicated, not through treatment, but by vaccine. And also, generally viral infections, they are much better, managed by the immune system itself. So and there are many others. For example, every time, for example, there's a drug that has been, proposed, it has been played down or blocked or canceled because of the procedures, that put in place in order to be able to allow, the official use of those drugs. So it is, the complex mixture of all these, situations that make people to, to doubt. We can see, for example, we've seen that many people have been vaccinated. I know people who have been vaccinated, many of them who have been vaccinated. But it's difficult to convince people. So so that's why it is important to have such conversation, such discussion, so that people can also understand, that, of course, there are conspiracy theories. People talk about, the vaccine containing, a microchip that might control people through the 5G and stuff like that. But, there's no real proof to that. And also, the prophecies in the Bible and stuff like that, people will talk about such things. But it is difficult to convince the amount of people when they have, conceived those things. But certainly, many people have already taken the vaccine. Millions of people, for example, in England have already, I think 2 million, as the last that I've heard, 2 million people and many other millions of people around the world have already been taking the vaccine. So probably in the next couple of weeks or months, then we'll start hearing if there is any, negative effects. We've seen people commenting about, the 23 people who died in, I think in somewhere in Europe, and some other people have died because of, after receiving the vaccine. But it's difficult to prove, as Dr. Harrison indicated, whether those deaths were direct results of the vaccine or there were other factors, that have pushed to that. So I think what I can say is that, we just have to educate ourselves and educate people so that we can understand, and people, the communities can understand that this thing is real and that people are really dying because of the consequences of these infections, and that we should understand that we should put our lives first, and then maybe our doubt, will come later. I mean, somebody was saying, we've seen many people, who are sick in the hospitals, or those who have recovered and who come and say, no, I'll be first to take the vaccine once they come because I don't want to go through this again. So it is such, discussion, and conversations that will allay the fears of people and probably help us to convince many people who were, in doubt about the vaccine. But
Thank you.
for now, it's only the conversation that will continue. Yeah,
thank you. I'm going to just give 1 minute, one last minute, people, please, to Professor Pascal Bessong to only answer one question of Professor Mayboom, the one to do with repurposing of medication. Why can't we use all medicine to cure this virus? There's hydroquinone, whatever stuff that Trump was trying to push. People, they were having backlash. There are other anti-parasitic medicines that were pushed. People are having problems with it. Why? 1 minute.
Well, Edwin, I think that was the approach. That was the very first approach to this pandemic. If you recall, I think as early as June last year, I'm from Oxford University, and they had a trial of a drug that was already there. This is an anti-inflammatory drug, and the reason is that, and the COVID virus, when it gets into the lungs, into the alveoli, actually it provokes an exaggerated allergy that actually results in an inflammation, and that is what actually worsens and somebody's condition when he's infected with SARS-CoV-2. And that drug had, I think, a 30% reduction in people who were not and severely ill. So that drug was already there and was being used. And I think most of the drugs that have been tested, and tried were all drugs that were known already and being repurposed, to assist in the management. But there's a limitation to which attempts to which you can throw your hands and pick up from the shelf what is already available. If you look at HIV, for example, as a history, the first drug for HIV, AZT, was a drug used to treat cancer that didn't work and was in the shelf. So I think drugs that were already known has always been one of the first port of call in managing a case like this. So this also happened in COVID. But as time goes on, alternatives that become much, much promising are dwelt on, and people want to channel their efforts and resources into use the things that they feel that has a more better chance of improving many people. So I think that's what I can say in this regard. But of course, as somebody mentioned earlier on, it is good to have these drugs in a trial. And in fact, we may have and the desire, so, let's use ivermectin, let's use this, let's use that. But the question is, have you tested it? Have you tried it? Again, who does a trial in the first place? Most governments don't do trials, frankly speaking, and the COVID-19 is giving us another impetus that a government can put money in vaccine design as well as production, as we saw in the United States now, as well as the UK. I would say also in the European Union. So that in the past was done by pharmaceutical companies, and these people do business. They produce a product to sell. Is what we forget. A drug for these companies is a commodity. They have to make their money back as well. So, and they want to put their investments in the place where they think they will have returns. So you see, we need to create this balance of who is doing the testing, who is doing the trial, and what are the outcomes, and is for who, and who can actually afford it. So I think that will be the response I can give to that. Thanks.
Thank you. Thank you, Pascal. Thank you to Samie Amidou. Thank you to Dr. Angela Harrison. Thank you to Dr. Ashwell Ndhlala. Thank you, everybody. We will communicate further if we are going to have another session next week. But this has been a wonderful experience together. The takeaway message is that the virus is real. The virus is killing our people. The conspiracy theorists, please, 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 if you really want to prove that indeed this is a conspiracy, please go and visit one of the hospitals. You will see. In Venda, we say, Liababa, Lina Mwadze. Thank you, everybody. Goodbye.
Thank you very much. Yeah, bye-bye.
Thank you, Edwin. Bye-bye.
Thank you,
Bye-bye.
Edwin. Bye.
Cheers, bye.
Thank you,
Bye.
panel.
Thank you very
Bye,
much.
everyone.
Edwin. Bye.
Bye. Bye.
Thank you. Bye.
Thank you. Thank you very much.
Thank you. Bye.
Yes, bye-bye.
Thank you, Doctor.
Thank you. Rutendo, thank you. You can close the meeting and share the link for the recording, please. Thank you so much.
All right. Goodbye.